Okay, last time we sat down with Dr. Bonnie Henry, BC's top doctor, to chat about the processes that go on in the back end when deciding how to communicate effectively with the general public during the ongoing COVID-19 pandemic. Today, we are so excited to talk knowledge translation with Dr. Pieter Cullis about the science that brought the mRNA vaccine to fruition.
Hello and welcome to Raincoat.
My name is Isabelle.
And I'm Sarah.
And we are excited to share how academia, industry, and communities can work together on complex problems and improve knowledge translation.
Today we have 2022 Canada Gairdner Award Laureate, former director of UBC's Life Sciences Institute, and a member of the Order of Canada, Dr. Pieter Cullis. And this is only the tip of the iceberg when it comes to his long list of accomplishments. So before we begin, Dr. Cullis, can you give us an introduction of yourself, of who you are and what you do?
Sure. I'm in the biochemistry. I'm a professor in the biochemistry department here at UBC. I'm one of the more senior members. I've been in the department now. I guess it's for over 40 years. I joined in 1978, and for a good proportion of that time, pretty much all of it, have been looking at or trying to exploit the potential for lipid-based systems, both for trying to understand their roles in biological membranes, but also to use some of these systems as delivery vehicles, both for cancer drugs and also for nucleic acid-based drugs.
So you developed the nanoparticles used in the Pfizer and Moderna vaccine more than a decade ago. What got you interested in the field of nanoparticles?
Yeah, so I'm originally a physicist, so my PhD is in physics, and when I hit the end of my PhD, I realized that the most interesting problems, at least the problems I found most interesting, were well outside the field of physics, and so I particularly in the life sciences, and I got a little bit interested in biological membranes, although I didn't know anything about them really. And I applied for at that time, the CIHR in those days was called Medical Research Council. I applied for a fellowship to go to Oxford, which I won, and ended up in the biochemistry department there, which was quite an experience. I didn't know what a protein was. I didn't know what nucleic acids were. I didn't know anything, but I knew a bit about NMR, nuclear magnetic resonance, and I used it to study biological membranes, and particularly the lipids in biological membranes, and I got really interested in the properties of lipids. Well, in order to study this, these lipids, we had the biological membranes were just way too complicated to get any clean answers out. And so we had to use model membranes, and this is where the lipid nanoparticles started to come in. But we also found that we could load cancer drugs into these lipid nanoparticles, and so that's what got us interested in the whole notion of delivery. Because we could package cancer drugs, most common anti-cancer drugs, into the lipid nanoparticles very efficiently. And so the idea there, of course, was to try to deliver cancer drugs much more specifically to where they need to go. I mean, it's a ridiculous situation we currently have in chemotherapy, where only 0.001% of the administered drug actually gets to the tumor, and all the rest goes and causes very nasty effects elsewhere in your body. And so we had this possibility for packaging and delivering cancer drugs. So we decided to start a company with the postdocs in my lab. So this is around about 1992. But anyway, we started a company called Inex Pharmaceuticals. And over the course, the CEO that we'd hired in the company came to me and said, well, I can't raise enough money, putting these old cancer drugs into these lipid nanoparticles. the gene therapy was very much in vogue at that time. And so we thought, well, how on earth can we encapsulate nucleic acid into a lipid nanoparticle by other means? And it turned out that in our studies of lipid asymmetry, we'd synthesized a lipid that's called an ionizable cationic lipid. And then it also turned out that these ionizable lipids were useful for getting into cells. And so a lot of this work we did in collaboration with a company in Boston called Alnylam. The end result was that the clinical trial was wildly successful. Mm-hmm. The results came out in 2017, and the drug was approved by the FDA in 2018. Well, by that point, we formed another company called Acuitas. And so we thought, well, maybe we can deliver much larger messenger RNA and have that, instead of silencing a protein in the liver, mm-hmm. Perhaps we can get a protein expressed in the liver. And so that turned out to be the case, that we could actually get proteins being expressed in the liver, and we got better and better at it. And then in about 2014, we were contacted by an immunologist at the University of Pennsylvania, a guy named Drew Weissman. And he wanted to try our system as a vaccine. In other words, putting messenger RNA in that's coding for a protein that's associated with a virus, and seeing if we could raise a good immune response to that virus. And so one of the first pieces of work was done with Zika virus, and essentially making mRNA that coded for the it's a membrane protein that's on the Zika virus outside on the outside of the membrane, and this worked incredibly well. And so giving complete protection against Zika viral infection. So this brought us to the attention of BioNTech in Germany. And so Acuitas started working with BioNTech, but that was on a flu vaccine. This is about 2018, 2019. And of course, in 2020, then all efforts turned to COVID-19. And BioNTech in turn had been working with Pfizer on a flu vaccine as well. Anyway, so this at that point they ended up choosing the lipid nanoparticle that we designed at Acuitas to incorporate into the Pfizer-BioNTech vaccine, of which, I think there was about 2.5 to 3 billion doses administered last year, and there's probably going to be 4 billion this year. So it's had a major effect in really controlling the global pandemic. But so let's went through all of that just to say that sometimes you start off doing quite different things, and they end up being sort of kind of basic research, but it ends up being incredibly useful in terms of a therapeutic. So that's the story of the vaccine, just at 30,000 feet and going pretty quickly.
Wow, that's a huge story. You talked so much about your journey with the lipid nanoparticles for the drug delivery. And, we are coming from a more clinical research background and having a lot to do with research on humans and rehab science. And in lay language, how would you define for us what lipid nanoparticles actually do?
Yeah, the simplest way to think of it, we have most drugs that we use are what you might term what's usually termed small molecules. They're 500 molecular weight or something in that range, like cancer drugs or drugs to treat atherosclerosis or whatever it might be. And the reason that we use small molecules is because it's only those molecules can actually get into cells, get across the cell membrane to affect some intracellular target. And so the larger molecules, such as nucleic acid-based drugs like small interfering RNA or mRNA, just can't get into cells. They're too big, too charged. And so, and also, of course, your body has very intricate and sophisticated mechanisms for making sure that they never do get into cells, because for obvious reasons, they don't want foreign DNA or RNA in there. That can cause problems that we're all quite aware of. Anyway, so and also they're very, very readily degraded in biological fluids. So they need delivery systems. So if you want to use messenger RNA, mRNA, as a therapeutic, you have to have a delivery system that is able to protect it from the biological environment, first of all, protect it from degradation. And secondly, once it reaches a target cell, to enable this cargo to be delivered into the cytoplasm of the target cell. And so that's really what the lipid nanoparticles do. We package up the mRNA in these 100 nanometer or smaller systems, and then they will then take that material, and say in the case of something being injected intramuscularly, those will be taken up into muscle cells and antigen presenting cells in the region of the injection, and cause and then the mRNA that's in there gets decoded into the protein that it codes for. Say the spike protein in the SARS-CoV-2 virus, which is the one that causes COVID-19, and then causes a very vigorous immune response to that protein. But the basic thing is, it's like a FedEx, if you want to think of it that way, that just protects the cargo, the mRNA cargo, and then delivers it inside the cell where it can actually be translated. So it's it allows us now to use much larger molecules as therapeutics, and we're not confined to the small molecule drugs, which are very hard to develop, take forever, and cost a lot of money.
Totally. I was just going to say, it sounds like a mail delivery service within your body. But I guess in the wet lab, or kind of the basic sciences area, I feel like I typically associate that with Albert Einstein and having this aha moment. So throughout your journey of discovering all these, the next big thing in lipid nanoparticles and all the related areas, do you remember having like a aha moment and what that process was like?
Well, I have to say it's a bit incremental. I mean, as I mentioned, right, in that long, basically it's going through the whole history of the story. One of the first steps was, well, how can we package these nucleic acid polymers like mRNA into a lipid nanoparticle? And if we in a non-toxic way, that there were these positively charged lipids that were available, but they were very, very toxic. It was certainly an aha moment when we managed to get these nucleic acids. We were using DNA at that stage, antisense oligonucleotides, but when we got those packaged in, and at say pH 4, and then raise the pH to physiological pH, and the oligos stayed in the lipid nanoparticle. They were encapsulated very efficiently. So that was certainly something where you say, wow, okay, now we've got these big things. If we can get 90 to 100% efficient encapsulation and all of that in very reproducible systems. The next step was to say, okay, well, now we've got something that's reasonably non-toxic and we can manufacture it, and is reasonably efficient at encapsulating our cargo. Does it actually work to do anything in vivo? And seeing that initially, our initial efforts were, there's some indications of that we were using siRNA at this stage, small interfering RNA, to silence the target gene in the liver. And we were seeing something, but the formulation was too toxic. In other words, we were operating at a level that a dose level where the formulation was pretty toxic, and so I went to a chemist, Marco Cipollini, in the chemistry department here, and said, well, how can we make our system a bit less toxic? And so he came up with a different variant on the ionizable lipid that we were using, and it was turned out to be not only less toxic, but it was also more potent. So that was certainly an aha moment, which gave us the idea that perhaps we could really optimize these systems to get really very efficient silencing or delivery to hepatocytes in the liver, and so we exploited that and ended up with systems where we had a therapeutic index of 1,000, which means we could give 1,000 times higher dose than the therapeutic dose before we saw any toxic side effects. And that's when you're developing a drug, that's always the thing that kills you is the toxic effects that drug may have. I mean, if you're aware from cancer drugs, they're often poorly tolerated, which limits the dose you can give, and so therefore their anti-cancer effectiveness. In this case, we had a big therapeutic window, so that meant we had it had real potential as a therapeutic. So that process became full of aha moments. You want to think of it that way, particularly as we got to this point where we had a formulation that was really not very toxic at all, but also highly effective. That's when we knew that we probably had something that might be useful in vivo, as in the clinic.
And how were you able to know that it was also safe? So when you made this discovery, you just talked a little bit about how you knew there is potential and that you knew it's something new and important. But how were you able to know the safety?
Yeah, the safety is partly this toxicity issue I mentioned. We're obviously, the liver is a target organ because we're silencing genes in the liver, and we had this window, very high window. We give 1,000 times higher dose before we would see, say, raised ALT, AST levels of the indicating liver toxicity. Now, in addition to that, you do the pathology to examine any other issues that might be arising. That came up very clean. The other part of this, of course, is with the RNA-based drugs. They don't get into the nucleus, so they don't affect the genomic DNA. They're only there in the cytoplasm, and so they're intrinsically extremely safe from that point of view as well. So the end result is what we knew we had the other part was we started to design some of the lipids so that they were readily broken down in the by esterases in the body, so that while they were there for the actual enabling the nucleic acid cargo to get into a cell, that they were relatively rapidly degraded subsequent to that. So these are pretty safe drugs for sure.
As our focus of our podcast is more on knowledge translation and how to really get your research out to the public or whoever your end user is. And in our case example of the COVID vaccine, once it found its use within the mRNA vaccine, that's kind of how it was translated. And just out of curiosity, is knowledge translation a common term used in the field of biochemistry, or is there another term that kind of encapsulates the process of translating that research into whatever it could, like the impact that it has?
Well, yeah, knowledge translation is certainly one way that's described commercialization, moving things into the clinic. There's quite a variety of terms. The basic thing, the basic thing is you're trying to move a discovery that's maybe made in academia into the real world and have it have some benefit, whether that's therapeutic benefit or other benefits. And it's a complicated process. And I've started up, over 10 startup companies over my career, and that's just simply to get things that are maybe have their origins in the lab, but to get them out there into the community. The companies I've started now employ about 400 to 500 people locally. So we're now known as a worldwide hub in terms of nanomedicine and whole lipid nanoparticle technology. And there's a number of reasons for doing this, in addition to the desire to get things into a, to be clinically used. One of the reasons I started doing this originally was simply to keep a team together. As I said at the outset, I had a very good team, and this starting a company was a way of keeping that team together. And if we hadn't done that, we wouldn't have achieved the success that we have achieved. You build up a reservoir of expertise and experience and know-how that is impossible to do in a long-term way in an academic environment.
And you speak of getting a team together as an important knowledge translation tool, and you show that with your companies. Would you say that also has been the way for you to collaborate with Dr. Carrico and Weissman before COVID-19 to get then the nanoparticles into the current vaccine? Or like, how did that knowledge translation process work in this context?
Yeah, it was a serendipitous event, really. We had the company called Acuitas, which I co-founded in 2009, together with Tom Madden and Mick Hope, were two of the original people in my lab in the early 80s. We've been working together for 40 years, but we were packaging up messenger RNA in lipid nanoparticles, and then and they're injecting that intravenously, and we were seeing gene expression in the liver. So that which was fine, but it had nothing to do with the vaccine. It's just that Drew Weissman read about this and said, "Boy, that's interesting. Now, if they can make cells in the liver, hepatocytes, make a protein using their delivery system, maybe it will also make proteins when it's injected as a vaccine. In other words, if you put in mRNA coding for a protein associated with the vaccine, associated with a virus, then perhaps that would make a good vaccine." And so we weren't going after vaccines at all. We were trying to make therapeutic proteins in the liver, which is a heck of a lot of therapeutic applications of, but it turned out that the application of pretty much almost exactly the same delivery system, where you have a messenger RNA coding for a viral protein, turned out to be a great vaccine when delivered intramuscularly. And the reason for that is not just the fact that you've got the protein being made in, say, muscle cells and antigen-presenting cells. This is where we got really lucky. It turned out that the system itself, the lipid nanoparticle, made a very good adjuvant, as it's termed, and so it was produced an immune response that was very, very potent. For example, the immune responses are often way better than you get by getting infected with the disease itself. Which is pretty incredible. Well, the 95% effectiveness speaks to that, with the original clinical trial. So yeah, it's you have to get lucky sometimes. We got very lucky with that one.
Totally. And I guess you mentioned commercialization is also a term used in biochem. And I feel like what you did with your lipid nanotech research and into the vaccines counts as commercialization. So was there anything that you weren't expecting to happen once your discovery and your science was implemented in something so globally implemented, such as the vaccine?
Well, when the results were announced in November 2020, as was mentioned, 95% protection, regardless of age, ethnicity, and the tolerability was pretty good. I couldn't believe it. I mean, geez, that's incredible. And for a vaccine that's 70% effective, that's considered pretty good. So this was really unheard of. So that's kind of one thing. I mean, you just say, wow, that's a pretty big deal. I guess the other part has just been the sheer impact. This has had worldwide. I mean, who could have could have ever prophesied that something you work on, you think it's important, but you never really think it's going to be that important. So this one turned out to be amazingly important in terms of from a global point of view. As I say, these are things you can't program. You can't say, "Gee, I'm going to do this. I'm going to cure a major disease and go at it in a linear fashion." You do have to have some luck along the way. That's for sure.
You mentioned earlier that you always work with your team together to build up your companies, and you work with industry together. But assuming that not everyone is in your field of work, I assume not everyone who was working with you in the company or invested maybe time and money always understood what you're doing. How did you deal with that? Or how would you even convince them that it's something important to invest time and money in?
Well, it's an interesting question. When you start a company, when you're, let's go back a bit. I mean, if you're just in a research lab, then of course your main thing is research, and you're trying to get some things, trying to understand biological phenomena and maybe get some things to work in a reproducible way. When you start a company, it really is. It's more of a team, because you need a team to do all the different things that are necessary to do develop a product. And so you need financial people who are going to help you raise the money and organize all that. You need management people who know how to set up an organization. Human resources. You need the marketing so much as people that will. It's in the early, not so much in the early stages, but you need people that are able to set up partnerships with other companies, so straight management capabilities, and you need legal. You need all kinds of different skill sets, and it's quite remarkable when you get a whole organization running like that that is all focused on the same end. Which might be getting a drug that's going to be a benefit to people. But I really enjoy that process. And it's actually not that, it's not, it sounds complicated getting all those bits and pieces working in a unified manner. But it's very gratifying when you get it so it's really humming. And it's one of the attractions of companies, actually, is that, extreme teamwork that's required.
Even before setting up this whole team, how did you pitch your project and your ideas to the people you wanted to hire? How did you convince them that this is important?
Well, this is some things you can do inside an academic setting. You have to have a pretty robust data package. I mean, say in the case of a cancer drug, for example, then you have to have some evidence in, say, an animal model of cancer that your particular formulation is working, so you get that together. You also have to have some intellectual property, so that it's clear that your product is well protected, and that there's nobody else can make it, and so you have that. So those are things that you have to have some idea of toxicity. If you can. Make the bigger the window, the therapeutic window, as I was indicating, the better off you are. The competing products, what else is out there? I mean, is there already a drug that cures the disease that you're trying to cure? That's not anyway. So you need to do those kinds of analyses in terms of the medical need, etc. But those are the things that I started up a couple of not-for-profit organizations that are really very much focused in that area, such as the Center for Drug Research and Development, which we started off in 2004, which is an NCE, a National Center of Excellence in Commercialization, and that's morphed into what is now ADMARE. So that's on the UBC campus here. It's in the same building as PharmSci. But they just got $92 million from the government to continue these sorts of this sort of putting together the full package that will convince an investor to invest in a particular technology coming out of a university. Those are the kinds of, it's those sorts of things that are needed to often make the data package convincing enough so that an investor feels assured that, there's a good chance this could actually work.
And I feel like we've talked a lot about the positives of co-founding a lot of biotech companies. So before all of our listeners start launching their own biotech companies, I want to touch a little bit on your learnings and potentially some challenges that you faced. So when launching Acuitas or any of your other biotech industry companies, what is something that you've learned in that process?
Oh, yeah, I mean, it's a rough business, right? I mean, luck or lack thereof can play a big role. One instance was with Inex company. We started off in 1992. We had a drug to treat leukemia, and so we ran a clinical trial, and then we went to the FDA, the U.S. FDA, and 2004, and we didn't get approval of the drug. They said they wanted some more data, and at that point we just didn't have the money to run the additional studies, and so we had to downsize the whole organization really dramatically. We had to let over 100 people go, so that was not a very pleasant situation. It also caused us to really pivot and say, okay, well, let's really go after the nucleic acid-based drugs. And so as opposed to small molecule drugs, which the cancer drugs were. And so in many ways that was, that ended up being quite successful, but there was that particular interlude that was really not much fun at all, where, you pin a lot of people's hopes to. We applied to the FDA just after Vioxx was. I don't know if you know the Vioxx story, but this is a drug that was approved for. I think it was a blood pressure drug, but anyway, it had really nasty effects, cardiotoxic effects in a small proportion of people, and so it had been approved by the FDA, and then they basically pulled it back. But they were in a very protective mood. So even though I feel that our cancer drug should have got approved in 2004, well, the vote went against it. And so that was that. So the vagaries of the approval process, the whole regulatory process that surrounds approval of a medicine it's a difficult process and very expensive too.
I feel like I'm seeing some similarities between academia and industry in terms of really being reliant on like grant funding or even funding in general. And it seems like money plays a big role in both aspects. So I guess one question for you is, you are very heavily involved in both academia and industry as a full professor and co-founders of multiple businesses. So what are some learnings that you've applied from the other aspect of your role? So for example, is there anything you've taken from the academic side that you took into account when launching your business, or learned something from your business that you applied back into academia and running your research lab, even in teaching classes? How do those two kind of different hats affect one another?
Well, it's a good question. Obviously the companies that I got involved in starting, it's science first, second, third, and fourth. That you really emphasize the need to be, that you really have to have something that's scientifically very valid. And it's amazing, but you end up doing often research in a company environments that is very important to do. That often you can't do in an academic environment, partly because of cost. To run animal studies, for example, at the scale that we had to get the necessary data to be able to go into the clinic. To do that in an academic setting, it was, just way too expensive. And so raising money so we could actually run that was an example of where it's research that you really find great difficulty in doing in the academic setting. The flip side is an interesting one. I've always tried to educate the students and postdocs in my lab as to the opportunities outside in addition to academia. People go through the academic process and and say doing a PhD or whatever, and really the only avenue that they see ahead of them is an academic one. And I think but the reality is, there's all kinds of different things that you can do with a PhD. Your employment opportunities are pretty much unlimited. And so and in the small biotech, you certainly have that. Those opportunities. In the early stages of a small company, I mean, every employee has to do everything, right? So it may be your expertise is at a bench level doing, particular experimental work, but you're probably going to be called on to talk to partners in terms of, setting up a collaboration. You're going to be talking to the lawyers about intellectual property. You're going to be trying to manage a small group. I mean, all of these things are talents and are vocations that people. Those opportunities are totally there for somebody in getting a PhD or doing a postdoc. So I really try to emphasize that don't feel limited by the prospects in academia. In addition, the salaries are a bit higher too. So, so another. Of course, that doesn't take much, but.
That's a good point. So last questions for today. You mentioned, I think, 2 key highlights for professionals from academia or people who work with complex matters. Is a key point is to have a good team and also really think outside the box to, for collaboration and your professional development. But do you have any other tips for, let's say, academic professionals who want to translate their knowledge at the right time to the right people?
I mean, the main thing is there's lots of programs that are aimed at trying to get academic discoveries out into quotation marks the real world. And so some of these translational grants, I mean, apply for them and start. The other thing is just to start stuff off. Sometimes you can imagine all the difficulties there are, and it inhibits you from actually doing something. I've always found, it's like ready, fire, aim kind of thing. You're never going to have the full picture where you're guaranteed a successful outcome. And so at some point, you just got to say, I'm going to go with this because it has the right feeling, and then just make it happen. So that's I think for a lot of academics, they get a bit scared by all the things they don't know, and not realizing they probably know more than they think. So.
I think the ready, fire, aim quote I've heard that recently from a lot of people. So I'm seeing a trend here, but I think that's definitely a great summary of what to do next. So to summarize, today we learned the whole timeline of how lipid nanoparticles, research direct from Dr. Pieter Cullis' lab here at UBC, has led to the development of mRNA vaccines that have been rolled out worldwide to combat COVID-19. And overall, it's important to remember to keep your eyes and options open beyond academia. And with a sprinkle of luck and collaboration, it could lead to a number of opportunities and discoveries, and you could potentially start some biotech companies along the way. So thank you again to Dr. Cullis for highlighting the science that really went into the vaccines and for a great informative episode.
That's it for today, right from the heart of Vancouver. Thanks to UBC AMS for supporting this podcast.
Keep in touch in the meanwhile on Twitter at RAINCOAT Podcast. Till next time.
Stay dry and stay safe.